Welcome to Click for IB Biology. We're studying diversity of organisms, and in this we're going to be looking at the unity and diversity of genomes. There is a PDF of this presentation that's linked in down in the description, and you can annotate that with your own notes. And then when you've finished with that, down there also in the description there's 10 questions. The answers are provided, and you can use that to test your understanding and comprehension of this part of the course. So we're looking at genomes. Now, it's possible for students to access a number of genomic databases, and I have linked two in particular into the description. The first one is this animal genome size database, and the second one is that associated with the Royal Botanic Gardens of Kew in England, in the United Kingdom. The first one rather, is for animals, and the second one is an excellent source of information about genomes of plants. And so I've selected a couple of very quick settings that I put into both of those databases to extract data. But first of all, let's take a look at the word genome. So the genome reflects the complete set of genetic information of an organism, and this includes both the DNA sequences and the RNA sequences. And we know today that RNA sequences are seen as very much the orchestrators of our genetic diversity at the cellular level and in the cell biochemistry. So very important for us to know about the diversity there as well. And so quickly I selected it from the chordates for your comparison within the settings of mammals, primates, family of the Hominidae, and then the species name of Homo sapiens. It was able to tell me the common name of humans and what is called the C value and the chromosome number. Now, the C value is effectively the genome size for the haploid gamete structure. So we remember that when we look at C value numbers, we have to double those to get a true value of the full genomic structure. The units are incorporated into the table that you can see when you access these. I then selected mammals, primates, hominidae, and from the same family, and the chimpanzee from that, and to take a look at the C value, and you'll be interested to see that the C value for chimpanzees is higher than it is for human beings, and that is the chromosome number is also higher, as we discussed previously in the previous video. Plants, you can do the same kind of selection there, looking up species of plants. You can see that I selected here the Sequoia sempervirens, which is the sequoia tree, and I was able to obtain the C value there, and you can see the difference, and you must be struck immediately between the C value here of 32 compared to that of the chimpanzee of 3.76. So that does raise questions in our own mind about how to interpret these databases and their uses. Links to both databases are in the description below. So by using those databases, I was able to extract a number of values and then plot them into this table for your consideration, ranging, of course, from Homo sapiens up here into all the way down through plants, roundworm, yeast, from what we use in bread making, and of course right down to the bacterial cells Escherichia coli. And here we have the number of base pairs, usually BP, and often expresses MBP, which is millions of base pairs, but you can see in fact up in humans it's actually rated in billions, and quite a range in values there for these selected organisms. The chromosome number is also selected here, and the estimated number of genes. Now, it would be worth pausing the video to see if you can find any discernible pattern in those chromosome and gene I took the information from the databases for these select groups. So using the animal genome size database and the Kew Garden one, what I established were a range of values for the C values. So these, of course, are extracted for species. And of course, there are very many species within the flowering plants and the groups which are listed here. So these are approximate ranges in the C values, which, if you remember, is the haploid genome in terms of base pairs that they contain. And so having done so, I then have created these bars to illustrate the range that we have and some suggestion of the complexity of the organisms as they, the evolutionary significance as they go up through the chart here. And the interesting thing, of course, is that when one compares genome size to the complexity of the organism, there appears not to really be any strict correlation. So it would not be strictly true to state that the greater the C value, the greater the genomic size, the more complex the organism. And just to highlight that, we can spot here, look at the huge sizes and range of the flowering plant C values, and we might want to think about why that is the case, which has actually been mentioned already in the form of polyploidy. But also down here in the amphibians, they show a tremendous range in their C values. And if we attempted to plot specifically humans on this diagram, I believe that they would be approximately here. And so attempts to create pattern between complexity and genomics doesn't really work out. And so we have to dismiss the idea that the size of the genome tells us about the complexity of the organism. That relationship does not exist, and in fact is referred to as the C paradox. So that's the takeaway from the comparison of haploid genome databases. Complexity and C values don't correlate. Genomic size doesn't correlate. I just wanted to, through this simple diagram, make one other point, which has been made in previous videos, but just to strengthen it, is the comparison between intraspecific variation and interspecific variation. So each of these plotted points here in this theoretical diagram represents a species. And you can see here that I have measured the mean body length of these species and plotted them as the mean plus a standard deviation and minus a standard deviation. And the purpose of doing so is to plot the mean and show the reliability of the mean, which of course is what the standard deviation does. One could change this to plus and minus a confidence interval that you could use for more direct comparison. But the point that I wanted to strengthen was that whilst there is variation within this particular species here, that if we then, and each one of the species has its own variation as shown, spread of the values around the mean like this. However, if we take the entire group like so, then its variation is far greater. So the variation intraspecifically is less than the variation interspecifically. The intraspecific variation will be to do with allele frequencies, and the interspecific variation, amongst other things, will include things like gene frequencies, the presence of other genes, and so on. We now have the link to the next video, in which we consider the application of genomics in evolutionary relationships and contemporary applications to health. The playlist is on the right-hand side, and the subscription button is right in the middle there for you to subscribe. I'd like you to do that. Thank you very much for watching this video.